Hi, my name is Arian. I'm first year Bachelor of Science in Nursing student. My video aims to help anyone to learn and an opportunity for me to contribute for my growth as a nursing student.
So let's try to have first an overview about our course, which is biochemistry. So biochemistry is the science in which the chemistry is applied into the study of living organisms. So this includes the study of atoms, molecules, and other chemical reactions which comprise the living organisms. So without any further discussion, we have these four principal types of biological molecules, or the biomolecules for short. First, we have the carbohydrates. Second is the lipids. Third is the proteins. And lastly is the nucleic acids. So let's go and discuss first about the carbohydrates. Carbohydrates are one of the most abundant and widely distributed organic compounds found on Earth. So they are usually ubiquitous, and they are being found as an important constituent of the plants and animals and the microorganisms. So carbohydrates are formed de novo in the process of photosynthesis. So in this process, it uses sunlight to combine the carbon dioxide and water to form the carbohydrates and the molecular oxygen, which is abbreviated in this reaction. So that is the abbreviation. So carbohydrates thus represent the conservation of the sun, the energy of the sun as a chemical energy, and serve as the major source of energy for non-photosynthesizing organisms. So therefore, we can conclude that they must have been very early products in the evolution of life. So next is we have the six major functions of the carbohydrates in the body. So first is providing energy and regulation of blood glucose. Second is sparing the use of proteins for energy. Third is breaking down the fatty acids and preventing ketosis. Fourth is the biological recognition process. The fifth one is the flavor and sweetness, or as sweeteners. And lastly is for the dietary fiber.
When a people eat a food containing carbohydrates, the digestive system breaks down the digestible ones into sugar, which enters the blood. And as these blood sugar levels rise, the pancreas produces insulin. Insulin is a hormone that prompts cells to absorb blood sugar for energy or storage. Protein sparing is the process by which the body derives energy from sources other than protein. So there are such sources like fatty tissues, dietary fats, and the carbohydrates. So using these alternate energy sources lessens the amount of amino acids that will be metabolized for energy. So our body will grab protein from our diet if it is available, and skeletal muscles and organs, and convert its amino acids into glucose for energy and to maintain normal blood glucose levels. So this can cause muscle loss, problems with immunity, and other functions of proteins in the body. Ketosis is a metabolic state where the liver produces small organic molecules called ketone bodies. So most cells in the body can use these ketone bodies as a source of energy. When there is a limited supply of external energy sources, such as during prolonged fasting or carbohydrate restriction. So these ketone bodies can provide energy for most organs. In this situation, ketosis can be regarded as a reasonable and adaptive physiologic response that is essential for life. So it enables us to survive periods of famine. When no carbohydrate is available, the liver will not break down fat completely. Instead, it produces ketone bodies that are used by most cells to provide energy. So when these ketone bodies are produced more quickly than the body needs, ketone levels build up in the blood, resulting in a condition known as the ketosis. So ketosis is most commonly caused by very low carbohydrate consumption or prolonged fasting. Adding a source of plant carbohydrates, such as simple sugars, especially during the initial stages of flowering, increases yields. So not only do plants utilize these sugars, but the beneficial microbes that helps roots with the nutrient uptake do as well. So some growers claim that using a flavor enhancer like pineapple or cane sugar-based supplements sweetens the taste and aroma of their harvests. So most plant sweeteners also contain amino acids, other component necessary for plant growth and health. Dietary fiber. So it is a type of carbohydrate that our body can't digest. So we should subtract the grams of fiber from the total carbohydrate. So on nutrition fact food labels, as we can see on the back of those junk foods, the grams of dietary fiber are already included in the total carbohydrate count.
So for the classification of carbohydrates, we have here the chemical classification of carbohydrates and the physiological classification of carbohydrates. So for the chemical classification, we all know that carbohydrates are also called as carbs, and they are defined as a aldehydic or ketonic compound with a small number of oxidrylic groups. So on the basis of the forming units, on the number of forming units, there are three major classes of carbohydrates. So that can be defined. So we have here the monosaccharides, the oligosaccharides, and the polysaccharides.
Monosaccharides, or simply sugars, are formed by only one polyhydroxyic aldehydic or ketonic unit. The most abundant monosaccharide is D-glucose, also called as dextrose. This table shows the classification of monosaccharides based on the number of their carbon atoms, their general structure, and examples for each. So, the three carbon atoms and these monosaccharides are called trioses. There are four carbon atoms, so these monosaccharides are called tetroses. So for the five carbon atoms, we have pentose, and for the six carbon atoms, we have the hexose, and for the seven carbon atoms, we have the heptose. The preferred structural form of many monosaccharides may be that of a cyclic hemiacetal. So 5 and 6 membered rings are favored over other ring sizes because of their low angle and eclipsing strain. These cyclic structures of this kind are termed Furanos, which is 5-membered, or Pyranos, the 6-membered, reflecting the ring size relationship to the common heterocyclic compounds furan and pyran shown on this video. The biochemists usually use two types of drawings called the Fischer projections and Howard projections to draw the sugar molecules. The Howard projections are sugar molecules depicted in their cyclic forms. So, these Howard formulas are convenient for displaying stereochemical relationships but do not represent the true shape of the molecules because it is a flat projection. While the Fischer projections are open chain forms as shown in this image. So the carbon atoms are numbered in the Howard and Fischer projection so that the edge in the Howard projection is one carbon atom. So carbon 1 in the Howard projection is also carbon 1 in the Fischer projection. Oligosaccharides are formed by short chains of monosaccharidic units, which is from 2 to 20 linked one to the next by chemical bonds called glycosidic bonds. The most abundant oligosaccharides are disaccharides. It is formed by two monosaccharides, and especially in the human diet, the most important are sucrose and lactose and maltose. Within cells, many oligosaccharides formed by three or more units do not find themselves as free molecules but linked to other ones, lipids or proteins to form glycoconjugates. Oligosaccharides also have many functions including the cell recognition and cell binding. For example, the glycolipids have an important role in the immune response. Oligosaccharides has a fructooligosaccharides or FOS, which is a dietary oligosaccharides found in many vegetables, is a short chain of fructose molecules. Polysaccharides are polymers generally called as glycans consisting of 20 to 10 monosaccharidic units. They differ each other for the monosaccharides recurring in the structure, for the length and the degree of the branching of chains, or for the type of links between units. Polysaccharides has also a limited water solubility. Polysaccharides are defined as homopolysaccharides if they contain only one type of monosaccharide, as starch, glycogen, or chitin. And it is heteropolysaccharides if it contains two or more different kinds like hyaluronic acid. For the storage polysaccharides, we have first the starch. So starch is the plant storage polysaccharide. It is an alpha glucan that has structurally distinct components called the amylose and amylopectin, and it is insoluble and forms starch granules inside many plant cells, but it is not a pure substance. It is a mixture of amylose and amylopectin. Next, we have the storage polysaccharide, which is glycogen. Glycogen serve as the secondary long-term energy storage in animal and fungal cells, with the primary energy stores being held in adipose tissue. So, glycogen is made primarily by the liver and the muscles, but can also made up by glycogenesis within the brain and stomach. So, glycogen is an analogous to starch, a glucose polymer in plants, and is sometimes referred to as animal starch. So we all know that glycogen is a form of energy reserve that can be quickly mobilized to meet our sudden need for the glucose, but the one that is less compact and more immediately available as an energy reserve than the lipids or triglycerides. For the structural polysaccharides, we have these two main types, the cellulose and the chitin, but we have also here the arabinosilans and the pectins. Cellulose is a fibrous homopolysaccharide of high tensile strength which forms a structural element of cell wall in all plants and in some fungi and protists. So it is also insoluble in water and it does not change color when mixed with iodine. And cellulose is the most abundant carbohydrate in nature. Chitin is the second most abundant organic substance. It is a complex carbohydrate of heteropolysaccharide type which is found as the structural component of fungal walls and exoskeleton of arthropods. And in fungal walls, chitin is often known as the fungus cellulose, and it is something that is soft and leathery. Therefore, it provides the both strength and elasticity. It becomes hard when impregnated with certain proteins and calcium carbonate. Arabinoxylans are found in both the primary and secondary cell walls of plants and are the copolymers of two sugars, which is the arabinose and xylose. And these arabinoxylans may also have beneficial effects on human health. The pectins. Pectins are a family of complex polysaccharides and they are present in most primary cell walls and in non-woody parts of the terrestrial plants. Acidic polysaccharides are the polysaccharides that contain carboxyl groups, the phosphate groups, and the sulfuric ester groups. So these acidic polysaccharides are known for their wide range of biological activities, and these natural products are obtained from animal sources or are prepared by the chemical sulfation of naturally occurring microbial and plant polysaccharides. And for the medical application or for medical applications, we have the heparin, which is the most commonly used acidic polysaccharide.
So, for the physiological classification of carbohydrates, it is classified according to the degree of polymerization. So, we have here the simple, the complex, the available, and not available.
So for the simple classification, we have the mono and disaccharides, which is also known as sugars, and tri and tetrasaccharides, which is oligosaccharides. And for the complex classification, it is simply the polysaccharides. A further classification lays the foundation and the possibility of being used directly for energy purpose. So we have the available classification. So as glucose, fructose, galactose between monosaccharides, sucrose, lactose, maltose, and maltodextrin between oligosaccharides, and the starch and glycogen between polysaccharides. For the not available classification, the members of this class, if ingested, are not digestible nor absorbable and will be fermented by intestinal flora with release of short chain fatty acids and so yielding some energy.
Chirality and handedness are a concept which is applied to the structure of molecules. So the word chirality is defined by the lack of features or certain features of symmetry of an object which leads to that object of not being superimposable of its mirror image. While the handedness is a different phenomenon that relates to the ability to classify the chiral objects to a right-handed or left-handed.
To know if a molecule is chiral, look for carbons with four different groups attached to identify potential chiral centers. Then after that, draw your molecule with wedges and dashes, and then draw a mirror image of the molecule. If the molecule in the mirror image is the same molecule, it is chiral. But if they are different molecules, then it is not chiral. A chiral molecule is a molecule that is not superimposable on its mirror image. So, if molecule 2 is not superimposable on its mirror image, and therefore it is chiral. And a chiral molecule is a molecule that is superimposable in its mirror image. Chiral objects have a handedness. For example, the golf clubs, scissors, shoes, and a corkscrew. Thus, one can buy right or left-handed golf clubs and scissors. Likewise, gloves and shoes come in pairs, a right and left.
In stereochemistry, stereoisomerism or spatial isomerism is a form of isomerism in which the molecules have the same molecular formula and a sequence of bonded atoms. So they only differ in the three-dimensional orientation of their atoms in space. So we have these two types of stereoisomerism. So we have first the enantiomers and second is the diastereomers.
So, in order to have a stereoisomers, the molecule must be chiral. Remember, in order to have chirality, molecule must have the following characteristic. So, for example, in this picture, the molecule on the left has 3 red hydrogen. Then these hydrogens are chemically indistinguishable from each other. So, the molecule is achiral. The molecule on the right has 1 red hydrogen and 3 other unique substituents. Therefore, the molecule is chiral. So for concept 2, the chiral molecules that have stereoisomers. Stereoisomers are molecules that have the same chemical formula, but differ in their arrangement at a chiral center. For concept 3, stereoisomers are identified by their handedness, which refers to the arrangement of the substituents relative to their importance. In general, elements of higher mass have higher priority. There are 2 types of stereoisomers, the enantiomers and the diastereomers. So for the enantiomers, it contains chiral centers that are not superimposable and mirror images. They only come in pairs. For the diastereomers, it contains chiral centers that are non-superimposable but are not mirror images. So there can be many more than 2 depending on the number of stereocenters. So in this image, you can see that there are 2 chiral centers. 4 stereoisomers are possible. Only the exact opposites, or the diagonal arrows here, are enantiomers, and they therefore have a mirror image that is not superimposable. The molecules with only 1 stereocenter that differs, the parallel arrows here, are the diastereomers.